Hi, this is Dr. Ben Morrill. Welcome to episode 92 of Reptile Genetics Weekly. Glad to have you here. It's already looking like it's going to be a fun year. Lots of good stuff coming. Specifically today, we'll talk a little bit. We realized when we went over albino back in the day, kind of like Ultramel, we said some about the test itself, but weren't able to didn't go over the specifics about what the mutation or mutations are that lead to albino. And we've seen over time, you all appreciate being able to see those details. So we love being able to show it and kind of educate a little bit about different types of mutations and what's specifically going on in these ball pythons that we all keep. And so, yeah, we'll talk a little bit about the specifics for albino, and then toffee and candy. That mutation is also in the same gene. So we'll talk about that mutation as well. And then, a little bit about business, what's going on, and things coming up. So good stuff. As far as samples, we have this week, I'm pretty sure we'll have results going out every day from fast testing. We had some interruptions last week because of snow in Texas. It's not very often in Dallas, Texas, they get, 5-6 inches of snow. Sean was out with his dog when I was talking to him on the phone. He has a puppy that's never seen snow before. I think was only about a year old and was experiencing snow for the first time. So, but that kind of shut things down in that area. But yeah, this week, everything looks good. We had some results go out yesterday and have a bunch going out today, Tuesday, and should be keep on going out the rest of the week. And sex determination should go out once or twice for ball pythons and colubrids. And then the panel results, I thought were going to come through over the weekend, did get delayed. There's a problem with the machine and probably didn't help with shipping things. And anyway, stuff got pushed back a couple of days getting our samples run. But I do have those results today. This is Tuesday. So I'm going to be analyzing them. I'm going to try to get through them as fast as I can. I just got them this morning. So there might be some results going out today, but probably tomorrow, Wednesday, will be when Wednesday and maybe Thursday at the latest will be when panel results are going out. So sorry, I fully expected that would be done over the weekend, but got pushed back. That also means that results for new tests got pushed back as well. So not only will I have the panel results that I'll be analyzing today and over the next couple of days, but the new test information I'll be going over as well. And it's beautiful. It's looking very good. Very, very excited about 2025. Things are going to be a lot different. Exciting times. But yeah, that's pretty much what I've got for sample updates. Kayla, how are you doing?
Hey, Ben, I'm doing good. I am so hyped about some of the stuff coming this year. Especially even in the first half of the year. You've hinted a little bit at me that we're not going to talk about this week, but oh my gosh, so exciting. Yeah. But yeah, doing well. I've got some really nice turtle pictures to show you guys today. That we're going to do business stuff, and then I'll show you the turtle pictures. Because we normally deal with snakes here, but this is very much in the realm of reptile genetics too. So, yeah. So getting business stuff out of the way. For those of you who are interested in getting into testing or testing, more than just a single gene in your animals, you can bundle them together with the bundles available on Clutch and Morph Market. They're a great way to save a little extra money if you want to test more than just 1 or 2 genes, and you also don't want to spring for a full panel necessarily. So highly recommend it. And we try to pass savings on to you guys when we can, because we, at the end of the day, it helps us too, because if we can do more tests all at once in a run, that actually makes it more cost effective for us. So the more tests we can do for you guys, the better.
Yes. And in these bundles, when you purchase them, the testing that way, those are all fast tests. So those will be one day turnaround, whether you do 2 or 5 or 8 or whatever, however many, those will all get done as fast tests. So that's the other benefit above doing the panel. The panel's nice because you get right now 30, and like we'll talk about in a minute, be even more here soon. But the fast test, the bundles, I mean, are awesome because you'll get those results fast.
Definitely. And let's see. So as far as sheds we're looking for, you guys have really been coming in clutch with sending these sheds, and we're still looking for more. I've seen some super vanillas coming through, and I think Ben mentioned that some blackheads and furrows have been coming through as well.
Yep, and Nyala and Stranger and RK Asphalt and
Oh
some
yeah.
specials and HRA and Mahogany.
Yay.
Quite
Awesome.
a few. We really, really appreciate those coming through. You guys are awesome.
Yeah, really appreciate it. We really can't do it without you guys, but this is part of our desire to complete our complexes or decomplex the complexes. That way we can offer you guys, like, if you want to test for the 8-ball complex or the spider complex, we can do the more niche things and also the main spread of genes in that complex. So thank you guys for those of you who have been sending sheds. And if you want to send sheds specifically for research, make sure that you don't have to send the entire shed. It can just be a little quarter $half size patch. That alone in that picture could be like 60 plus samples for us for what we do. It really doesn't take a whole shed. And send, of course, the species and the animal ID and name, any known morphs, any possible morphs, who it was produced by, and any lineage information. That can help out, especially if we find something strange that we want to dig into more. You've seen some of those happen here on our show, because we end up in some interesting rabbit holes, even when we're not looking for them.
Yeah.
So all this info helps us help you. But yeah, so we mentioned the spider complex testing in mega panels last week. So I know with the snow delays, we haven't been able to move as fast as we've been hoping, but any updates on that, Ben?
Yeah, not any huge updates yet. Hopefully next week, but yeah, that spider complex will at least have 2 or 3 or maybe 4 more tests in the spider complex.
Ooh.
And I would imagine sometime in February we will have a spider complex test. It will be a longer turnaround, like the panel test. It won't be like the bundles or the fast testing. It'll be more like the panel test, where it'll be like 3 to 5 week turnaround, but it'll be very affordable to be able to test for all 10 or 11 or 12 mutations in the spider complex. So if you're wondering if you have something in the spider complex and are really only interested in testing those, but not sure which one or ones you've got, then that test will be a really nice one.
Awesome. And likewise with the big mega panel. So how are we looking with that?
Yeah, that too, I think we'll have available in February, and that will be nice. Once again, the brand new tests we'll be coming out with over the next little bit won't be on that, because it takes a little time to add brand new tests to the panel. But the ones that have been available for the last 3 or 4 or 5 months that aren't in the panel, those ones will become part of it, and it'll be a much larger panel, a lot bigger than 30. So that'll be nice. And like we said last week, as a little bit of a genetic testing hack, is if you think you're going to need panel testing, if you pay for it now but use it once the larger panel is available, like I said, hopefully sometime in February, it will, even though you paid for it at that 30 morph test, when you turn it in, if we have more than that on the panel, you'll get all of those run at that cheaper price. So if you're going to buy panels, it's a good time to get them now before it gets bigger. And obviously the price will have to go up some, since we're going to be putting significantly more tests on that panel. So
yeah,
get them while you can.
Get them while you can. And when you do start adding on the extra, like when you are able to add new tests onto the panel, are we going to let people know here when
sure.
that happens? Okay, awesome. Sounds good. So be watching for it, y'all.
Yes, yeah. When the spider complex test is available, and when the larger panel test is made available, we will definitely let y'all know.
So exciting. All right. So turtle pictures. For those of you, so for some preamble, I in addition to working for Rare Genetics, I also work at a wildlife hospital. And one of the animals that we work with every single day are eastern box turtles. And since I do social media for them, I decided to just hunker down one day this winter and just I made a light box out of cardboard and copy paper and a glue stick, and I took pictures of every single box turtle patient we had. And the variation in this species, which is threatened in Virginia, is just amazing. Of course, a lot of them, you can tell they've been, hit by cars or lawnmowers. They have all kinds of injuries because, we're at a wildlife hospital, but they're amazing. And I wanted to show you guys because this is just the variation in this species we have in our backyard out here. It's so cool.
How many counties, is this in one county or in 3 or 4? How spread out are these coming from?
Oh, all over Western Virginia. These guys are this represents just a
big Virginia.
Yeah, all over Western Virginia and Central Virginia. Usually we don't get from the eastern part of the state, but weirder things have happened. But yeah, no, they're just so cool. And let's see, the one in the middle here, this middle section on the far right, that's the turtle that I found in a road. So that's quote unquote my turtle. Not my turtle. He's, he belongs to the wild, but that's the one I pulled out of the road.
Really pretty pattern on that one.
Right. There's one, this one middle second from the left. I love the starburst pattern on him.
Yeah.
They're so cool. And of course they are imperiled in Virginia, which is really sad because of poaching, but habitat loss, and honestly just people just taking them out of their backyards and having no idea how to care for a turtle. It's a huge definitely a huge risk to them. So every single turtle that goes back to the wild really, really helps out their species. So really love these little dudes, and it's a treat to work with them at the center. So just wanted to share how amazing their natural variation is with you guys. Because yeah. And I feel like something about how we are in the reptile community, I'll look at an animal, the way we look at the variation in our own reptiles, we kind of have a different eye for it, I think, than the average person. And so, I don't know, I feel like you guys might really get an extra appreciation for that, for just how amazing they are.
Yeah, that's a really nice layout. You can see very clearly side by side just how much variation there is. That's really cool.
Yeah. And the one middle second from the right, big round girl, I call her Big Mama because she's huge. It's like a big old hamburger of a turtle. And love her heart. She was hit by a vehicle, and so her you can see that disfigurement in her shell that's grown over, but she is you can't really tell from this picture, but she's almost a melanistic version. There's only a few little bright patches, and then the rest is just all dark. So cool. Yeah. Turtles. So let's get to the meat of this episode and do our monthly-ish morph spotlight for the albino / candy / toffee mutation. So Ben, I'd say we'll take it away.
All right, so we have shown you a little bit from this picture. The most recent one was ultramelt. We also went through lavender albino and the specifics of how that mutation, that's a single mutation. It's a large deletion that gives us the lavender albino. So yeah, today with albino, this once again is from the Eastern Michigan group. So this is Dr. Hannah Seidel and her students at Eastern Michigan that did this work, and it got published in 2022. So it's kind of crazy. It's coming up on, I guess it's a little over 2 years. It's crazy that much time has gone by. It seems like it just out, but
man, that's wild.
Yeah, very, very cool publication. Very thankful they did this work, and they did a beautiful job with it. Let's see. So the gene that we're looking in that will be represented by TYR. So that's the gene where the albino and the candy / toffee mutations are. And so you can see here the candy slash toffee that got proved to be the same mutation,
Mhm.
just two different lines that we had as breeders. But the candy slash toffees have a lot more of like a lavender. It's not really a white background. It's more like a lavendery, caramely background instead of the
Like a
white.
butterscotch kind of coloration as the background.
Yeah,
Yeah.
pretty
Okay.
cool. And then
Mhm.
the animals that have, because this is in the same gene, the albino, both the albino mutations and the candy slash toffee. So if you have an animal that has one of the mutations for albino and one of the mutation for candy slash toffee, you get what's called a candino or a toffino. So it's kind of, when it right comes out of the egg, it's hard to tell for sure which of the babies are albinos and which ones are toffinos or candinos. But as they get some age, they are kind of like in between an albino and a toffee or a candy. So that's
Ooh.
pretty cool too, that they do act like a super. They are compatible, but that double heterozygous animal that's got one mutation for albino, one for candy slash toffee, does look a little bit more in between the homozygous for either one. So that's also kind of interesting. Not something we see. We see it in other reptiles, but specifically in ball pythons. This is kind of a rare situation for ball python mutations.
Yeah. So let's see. This is figures A and B. So what is happening here?
Yeah, so this and we kind of worried from the beginning that if we put stuff like this up that we would see a drop off, but we've actually seen you all that come specifically to watch this show know that this is the kind of stuff that we like, and it's something you like too. So we're very thankful that you all appreciate this information. And so this is kind of a fun one. So this is the TYR gene that right under the A. Once again, the boxes are rectangles there.
Mhm.
Those are showing the exons, or the part of the DNA code that codes the amino acid sequence or the protein that will eventually be produced.
Mhm.
Uh, so the lines in between the boxes, those are our intron, or what we used to call garbage sequence, but it's non-coding sequence that sometimes does play a role in other things. And then after that first exon, you see that little arrow above. That's to signify where the promoter is for that gene. And
Hmm.
promoters, sometimes there are changes in promoters that make a difference in expression, or how something works. So it's cool to know where those promoters are if it's a gene you're interested in. So after that first, the promoter, and then you have that first rectangle, you have two slash marks. That means that intron is really long. And so they're just shortening that down for you. The rest of those introns are to scale. So you can see there can be quite a bit of sequence in between the different exons, which is just how genes work, which is kind of
Yeah.
interesting. Every once in a while we'll get a gene that is just all continuous. There's no introns at all, but that's fairly rare. A lot of them look like this TYR gene, where you have multiple exons that are spread out with pretty long sequences of intronic or non-coding sequence.
Hmm.
And then right below that, you see that haplotype. So this is a cool and interesting thing, and is something that we still don't have any publication that shows the most common albino mutation. So we see this, I would say it's 80-90% of the time when someone puts a albino in for panel testing. So we
Hmm.
know it's an albino, it's a visual albino. It'll be homozygous for this first or kind of more complicated mutation that we don't know for sure. We're just going to call it today the larger mutation, because it's not just a little snip.
Oh.
But to be able to analyze the animals that looked like albinos, but they couldn't figure out for sure what the mutation was. We're, like I said, going to call this larger mutation, will be in red. You can
Mhm.
see in that second graph where they have the red, blue, and green, that by far the most albino animals they tested were were that more complicated or larger mutation. And that haplotype part, those are SNPs near that gene, and those are ones that that by looking across all of them, you can kind of see which ones are most likely to travel with this larger mutation. So they're just trying to figure out a way to be able to tell if an animal was albino, even though they didn't know for sure what the mutation was. And so it's kind of a more complicated way to look at a sequence and figure out whether it's likely het albino or not. And thankfully, we've figured out a way to be able to test for that works well. But it was definitely at this time when they were publishing this a complicated thing to figure out for the majority of the albinos. Now, the one in blue, this D394G, so that's a D, the amino acid, changing to a G amino acid. Just from that SNP, that one ATCG, that changes an amino acid. And that's the less common albino mutation. That one's a SNP. That one is in the publication, how to test for that. So that one's really easy to test for. And then the Toffee candy, that bottom one in green, that's P384L. That's once again a P amino acid changing to an L amino acid because of that one base pair change. That one is also easy to test for, is in the publication, and we've all been testing that for a long time. So that's what's going on here. And then on the right side, the B part, what they're trying to do is just look across a bunch of different animals, and this part of the sequence where the D394G and the P384L, so the less common albino mutation and then the Toffee/candy mutation, they wanted to look at across a whole bunch of species and see, okay, if this mutation we know is occurring in ball pythons, how common is it that this same mutation is in other things? And so if you look across a bunch of animals and it's always a P, so like for the 384 position, you can look in shark, fish, frogs, human, chicken, turtle, lizard, a couple of pythons, it's always a P. So that's a very well conserved amino acid. So it seems like if you can look across that variable, that wide of a group of animals, and it's always a P, then that's a sign to you as a scientist, like, okay, that's really important for this protein. If it changes, then that animal, either doesn't live or isn't born or whatever. So that P is very well conserved. But in the ball python, when it gets changed to an L, then we get an albino. And then the same thing when you look at 394, that D is very well conserved across all those different animals. But in ball python, when it changes to a G, then you get an albino. So that's just a cool thing that they did to be able to show that those two positions are very well conserved. And then in the ball python, when that change occurs, you get either candy/toffies or you get albinos. So that's pretty cool.
Yeah. I'm glad you explained the going back to the colors on where it says TYR and then the colored D394G. I always wondered what those meant, but that's easy shorthand to show what it normally is. Here's the line in the protein sequence and what it changes to. Okay, that makes much more sense.
And that number, the 394 and the 384, that's the actual amino acid. So if you look at the TYR protein and you look at the whole protein, so the amino acid sequence, that's different. There's a DNA sequence that codes RNA that makes the amino acids. So it's 3 different sequences. But if you look at the amino acid sequence, that's what they're showing here, this D394G. That means if you look at all of the amino acids that make the TYR protein, if you count 394 out, that's where you can get an albino. And then if you count 384 out, that's where you can get a Toffee/candy.
So interesting. And very cool that those two amino acids are so common and so apparently vital among vertebrates.
Yeah.
It doesn't look like there are any invertebrates, but that's fine. Albinism has been pretty heavily studied in vertebrates specifically. So very cool, and it makes me wonder. It makes me think of like the Anole study where they used CRISPR editing to change to create an albino anole lizard. It makes me wonder if one of these amino acids were the ones that they edited to create an albino anole.
Yeah, and I'd have to look at that publication. I can't remember. There are multiple genes in vertebrates that can make it look albino.
Yeah.
I don't remember for sure if it was the same gene, the TYR gene, but it could have been. But yeah, it very well could be. You can see in this several, like the SANDP, all of those are well conserved. But then when you get to 385, you can see in frogs and then pythons, it's a V, where in all the other animals, it's an I. And you have a well-conserved F, and then you have some variation in 386 and 387. So yeah, there's some variation in this region, but you can tell that, SANDP, those are all important and shouldn't, if there's a change there, it's probably going to have a significant impact. And maybe changing the SAN or D, any of those might also make an albino because it's well conserved. That's a possibility.
Yeah. Oh, so interesting. Okay. Thank you for explaining that. So here's figure C. So what's going on here?
So here they're looking at breeder designation. So if the breeder sent in the shed as an albino, that's what that top line is. Okay. So when a breeder sent in a shed as an albino, they're showing that it was homozygous for that larger albino mutation, 25 of them, versus 8 where they have one copy of the larger albino mutation, one copy of the SNP, and then just 2 that were homozygous for the SNP. So you can see, just like what I said for us, it's like, I don't know, 80, 90%. Here, going out of these animals, 25 were homozygous for the larger, only 2 were homozygous for the smaller. So you can see it's in their population, they looked at that larger mutation is the more common one.
Yeah.
And then there was one animal that was sent in as an albino that was actually a Candino. That's why it's that albino and green P384L. So that breeder would have got to find out that, oh, that one I thought was an albino is actually a Candino or a Toffino. So
how about that?
Cool. And then one sent in as Candinos. 3 of them were actually just straight albinos. 5 of them were Candinos. And then with the larger mutation as the albino part of it, and then one of them was a Candino with the SNP albino mutation. Ooh. So that's where those numbers come from. And then the one that was sent in as a Toffino was a Toffino that was the larger mutation, was the albino part of that Toffino. Then the one sent in as Candy, there were 3, and the one sent in as a Toffee was 1. All 4 of those were homozygous for the Candy/Toffee mutation. And then for the non-albinos, there were 56 of them, and out of those 56, 2 of them were actually heterozygous for a mutation, but the breeders didn't know that. One was heterozygous for the larger albino mutation, and one was heterozygous for the Candy/Toffee mutation. And so those breeders would have got a surprise too to find out, oh, I didn't know that one had Candy or Toffee in it or albino in it, and actually it was het for that. So pretty cool.
Surprise. Yeah. Cool.
So that's from like a scientist standpoint, that sample size, that last picture, we put that back up again. That's important as a scientist to know, okay, did they test, 5 albinos? Did they test 25 albinos? Did they test 500 albinos? The larger that number goes up, the more likely that their conclusions are correct. So here, where they ran, it looks like 35 albinos, or I guess no more. It would be 38 albinos and 56 non-albinos, and then some hets. That's a pretty healthy sample size. And of from what we've seen in running samples on panels where customers tell us whether they think it's an albino or a candy or a toffee or none of that. This seems to be even after running thousands of them, this seems to be telling the whole story as far as we can see.
That's awesome. So here's figure E.
Yeah, so with this, it's just kind of blowing up that what we talked about before. You have that promoter, you have the double dash line saying that's a really long intronic region.
Mm-hmm.
You have the second exon and then the third exon. In the third exon, you have both the D394G, that's the candy toffee mutation,
Mm-hmm.
or that's the SNP for albino, and then the P384L is the candy/toffee. So those are both in the same exon, so they're really close to each other. So out of all of the sequence that has to do, you can see on the far right, 5 kb, that's how long 5 kb is. So, this is a pretty long part, but that exon is a very small part of that 5 kb. So it's a very small exon, but it has where both the SNP albino mutation is and the candy/toffee mutation. So they're very close in the same gene. Whereas the albino mutation, they did a whole bunch. If you look at down at the bottom, the larger albino mutation, all of those gray rectangles on the bottom that are tiled across, those are different PCRs they did to be able to try to figure out where this other mutation was. And they couldn't figure out exactly what was going on, but they did figure out kind of in the middle there. So a little bit after exon 3, somewhere in the intron, they do see an interruption or something that's different. And that's where that star and the two arrows that are pointing towards each other. So that was a primer set and a region that did seem to be different in the ones that were het for albino versus not. But it wasn't like a clean, easy thing. They could figure out exactly what it was going on. So that's something, hopefully in the future, we'll be able to get a publication from them or someone else where we find out exactly the detailed reason why this main albino mutation, why it was difficult to see back then, and exactly what it is that, like I said, it's like 80% of the samples we get have this larger, more complicated mutation.
Interesting. So now, after we've been testing it the way we have been, do we know more about where that is, or is it just kind of like we have the test, we know it works, it just works kind of thing?
Yeah, we do not have a detailed, I don't have something I could publish right now about the detailed part to that larger mutation. Yeah. But we do have something that does work. And like I said, we've tested it in thousands of known albinos, het albinos, and non-albinos. Yeah. And we tested, whenever we run an albino test, we run both the D394G SNP and we run the larger albino. So we know if someone turns it in as an albino, we always get homozygous for one or the other, or het, one of each.
Right.
And yeah, so it tests well, and we just don't have that detailed work. That's something that we're very thankful the people in academics are able to take the time to do that, and hopefully they'll get to that with this albino mutation before too long. But yeah, it's something so far we haven't had the time to, because we just need a test to work, and we're going to design more new tests. Eventually, if the academic people don't publish it, then when we slow down and making new tests, then hopefully I'll get to play with that and figure out some of these that are not completely detailed out yet. And really in 2025, I do expect us to have some publications come out. There's going to be some pretty cool ones that we'll have. And so it'll be the first time that we as RGI are doing some of these publications. So far, we've talked about tests we've developed, but not the exact details of, what it is, what gene, and what's changed. Or we've talked about things that other people have done in academics. But by the end of 2025, we should have some where we did the work and we know what gene it is and what the specific change is and all that stuff. And so those will be even more fun videos because it'll be work that we did.
Yeah. Oh, well, that'll be cool. All right. So as discussed, the albino or the albino trait, has 3 mutations associated with it. That larger albino that we've been referring to, the albino snip, and the candy toffee snip. So how does RGI grade the tests that we use? I'll leave that to you, Ben.
The 2 SNPs, the albino SNP, which, like I said, is the less common albino mutation, but it's the easier one to test for, and then the candy/toffee SNP. Those ones are in Dr. Seidel's publication, and we've done many, many times. So those are A+. And then the larger mutation is only an A, just because, like we've talked about before, we only give it an A+ if it's been published. Something to get published in a scientific paper, that means someone did the work to write it up, and other people that weren't part of the study are then looking at what they wrote up and giving them criticism and figuring out if what they've done was enough. And so there's not only the person doing the work, but there's other scientists looking at that and reviewing it. It's called peer reviewed, reviewing it to make sure. So that's why we put the plus on when there's been that extra amount of work that's been done. And so hopefully, like I said, someone in academics will get that done, or like I said, eventually, if it's us that gets there first, then we'll be happy to do that work and get it published. But for now, it's an A. So the SNPs are A+, the larger mutation is an A.
Very cool.
Yeah.
And of course, we had a lot of people contribute to this this study all the way back in, I guess it would have been 2021 probably by the time they were sending them in.
I bet they're sending them in for 2 or 3 or 4 years even before. So some of these might have gotten sent in even before 2020. Oh
yeah, I would not be surprised. But yeah, lots of familiar faces there. It's from back when Justin Kobilka was still Kobilka Reptiles instead of Canova.
Yeah.
So way back in the day.
Thank you to everyone on this list, and those of you that continue to send sheds in, to any of us that are working on ball python morphs and any other reptile genetic stuff. That's extremely helpful.
Yeah. And so speaking of showing some gratitude for sending sheds, our shed donor shout out this week is Barbarian Creations, or Samantha and Craig of Barbarian Creations of Wisconsin. Words are hard. But they saw our little call for more for like specific ball python genes, and they came in clutch with the NOCO special line, which we've been looking for. Redhead, more lace, which is super useful because we're trying to get that working as well. And so we're just really grateful for you guys. For
Redhead and Trojan, those are ones that are going to be good to have eventually as well.
Yeah. And they attend shows. They definitely have some cool stuff on their Instagram. Like I got this picture of some cute little hatchlings all piled up together. You still have like their fresh, like haven't even had their first shed yet. I love those. And some of the animals that they have currently listed on Morph Market for a steal, actually. So if they've got, looks like a possible blackhead in there, which and of course the het red axanthic, your spiders. So definitely worth checking them out because they've really been helping us out with our research into developing more morph tests.
Yes, thank you very much. It's very much appreciated.
Yeah. But all right, that's all we have for today, you guys. So just make sure that you like, subscribe, and comment. Let us know if these like deep dives into the let us know if you like these monthly-ish morph spotlights, or what you thought of the turtle pictures. And if you're sending in some sheds for us, let us know because we are definitely looking forward to those. So
yeah, and we are starting to look at shows that are coming up. And unfortunately, I won't be able to go to. There's one in Atlanta next month that I'll unfortunately miss again. One that Bob's Balls, one that he's running. So let us know if you're going to that one. I know you said we just got a Klubrid Fest date in September, so that's still a ways out. But let us know if there are any shows that we haven't been to yet that are good shows. Let us know in the comments. We can start planning. We're looking ahead now, seeing when things are happening and getting those plans in place. So let us know what you're going to and what we've missed out on if we haven't been there. And if it's one we have been to, tell us we should come again if you're going to be there.
Yeah, definitely. All right. Well, thank you guys. And we'll see you next week. Outro in 3, 2, 1.